I'm Win Arias, a senior scientist emeritus at the National Institutes of Health in Bethesda, Maryland. And it's my pleasure and excitement to welcome you to what is the third session in the 24th year of a course called Demystifying Medicine. By my calculations, this is the 456th consecutive session of this course. The goal is to bridge the exciting developments in biology, engineering, and computer science with medicine and global health. This is really a course in bridge building. The logo, shown in the upper right, is the Brooklyn Bridge. And today, we are like the individuals on that catwalk, sharing information, learning from two world-class experts in the topic of today's consideration. Hopefully, this kind of bridging accomplishes in a course similar to what happens in a real bridge. In the beginning, people may have misunderstandings about what each side stands for. And to do the bridge takes many components, not just one. But one thing is for sure, if you establish this kind of connection and scientific link with medicine, both sides benefit. So this course meets every Tuesday from 3 to 5 Eastern Standard Time. It's all virtual during this 2026. The link for watching it is shown here. And for those seeking CME credit, the code for today is 67266. Now, very important, please submit questions during the speaker's talks. And you do that by clicking the Send Live Feedback on your videocast display. After the two talks, we will have an active exchange of questions and discussion. So we urge you to do this so you can participate. Note also that all previous sessions for 24 years are in the NIH video archive and can be downloaded at any time. The goal, as I've said, is to convert silos of information, the expertise that sits in specific areas of science, basic and translational, and to convert silos, in effect, into a bridge. Next slide, please. Now, today's topic is HIV/AIDS, a persistent pandemic, despite the presence of effective treatment which prevents transmission. Now, for those of you who weren't around professionally 3 decades ago, permit a very brief highlight of events leading up to today, although the topics I will just pass by are going to be discussed in great detail. But 3 decades ago, those of us who, like myself, were making rounds in big city hospitals, particularly in New York and San Francisco, were seeing large numbers of young men dying of infections which normally from organisms that are normal components of our flora, but now they had turned into a death rattle. That disease, epidemiologists told us at the time, was prevalent in a group of Haitian immigrants, in heroin users, in homosexuals, and in a group of individuals who had hemophilia and were being treated with multiple blood products. This was known as the 4-H disease. Nobody knew what the agent was. Bob Gallo at the NIH had described a retrovirus causing a form of malignancy, and he and others around the world, particularly Luc Montagnier in Paris, proceeded to do classic work that eventually led to the identification of the HIV virus, or retrovirus. And Montaigne and his colleagues received the Nobel Prize for this. After years of study, information emerged as to the possible routes of transmission and the routes of infection, and even the mechanisms of disease were established. Remarkably, the total deaths from HIV/AIDS as of last year is over 44 million people around the world, and a large number in the United States. At the present time, there are still 40 million people who are living with HIV. The remarkable discovery that antivirals could reduce the viral load, preventing transmission and AIDS, permitted individuals who take these drugs to live a relatively normal life, some living as long as 50, 60 years, whereas before, HIV/AIDS was a death sentence, often within the 20s. So there are many thousands of new cases each year, here, globally, and there's increasing with the loss of aid, PEPFAR, and other programs, particularly in sub-Saharan Africa. Next slide. So today we have next slide, please. Today we have two world authorities to discuss, on one hand, the clinical disease produced by the virus, and where do we stand in terms of its control. And to discuss this, we're honored to have Carlos Del Rio, who's a distinguished professor of medicine at Emory and also in public health, and is a co-director of the Center for AIDS Research at Emory, and also heads the CDC HIV clinical trial. He's participated also in vaccine studies and others. Carlos has originally got his degree in medicine in Mexico, came to Emory for a residency in medicine and infectious disease, returned to Mexico, where he became executive director of the National AIDS Council for the entire country, and then returned again to Emory, where he was chief of medicine and also of the Global Health Department. His research is on HIV prevention, care, and to translate to the results of basic science into patient care. He's been very active with the World Health Organization also in influenza pandemic. He's a member and director of the International Antiviral Society. At the USA, he has chaired the PEPFAR Scientific Advisory Board. He edits the HIV/AIDS sections of the New England Journal of Medicine Journal Watch, has published it extensively. Carlos has received many honors, including from the National, the Nakomi Citation, Creedmoor Achievement Award, an election to the National Academy of Medicine, and in 2020 was made Foreign Secretary. Our second speaker is well known to demystifying medicine. Next slide, please. John Coffin has spoken at least 9 times in these sessions. May I have the next slide, please? Come on. Oh, what happened? John is at God damn it. John is at Tufts University, where he is a distinguished professor of molecular biology. For a period of time, he was active at the NCI, where he was the former director of the HIV Dynamics and Replication Program. and a special advisor to the Center for Cancer Research. He's a member of the National Academy of Sciences and is also a recipient of an American Cancer Society lifelong professorship. John is an outstanding basic virologist whose main concerns are the molecular biology, virus-host relationships, pathogenesis, and evolution and population dynamics. He's made continuing major contributions to understanding this ubiquitous virus and why we don't seem to be able to truly get rid of it. So Carlos, would you please begin, and thank you again for being with us.
Thank you very much, Dr. Arias. Can you see me now?
Not quite yet.
I don't know why is it not turning on. Okay, thank you very much. Thank you very much for the opportunity to join this and to speak to this group. What I will be doing today is telling you a little bit about where we are in HIV prevention and where do we stand. So, antiretroviral drugs, the history of the approval of antiretroviral drugs really starts, as you mentioned, 1981, the emergence of HIV/AIDS in the United States. By 83, the virus was isolated, but it didn't take us until 1987 when the first drug, the first nucleoside reverse transcriptase inhibitor, a drug named cytobuline, or ACT, was approved and was shown to approve as monotherapy. After that, there was a lot of discussion. You could not necessarily, most people started treatment and then eventually succumbed to the disease. So it was very clear that we needed to do more. And, in virology, typically drugs were not combined, but a big change was that virologists agreed that we needed to try combination therapy. And it really was in 1996 with the combination of multiple drugs, what we at that point called HAART, highly active antiretroviral therapy, that for the first time we were able to see the virus being completely suppressed and people no longer die of HIV, but actually remain alive. So that was really a watershed moment. From then, development of new drugs continued to occur. And really another major development was development of single tablet combinations. And nowadays, really, most of the drugs we use for treatment are single tablet combinations, whether it's in the US or globally. But improvements in therapy have continued. And the next big development was really the development of long-acting agents, and the development of long-acting agents with the idea that you can give either an injection or why not eventually also a pill that you wouldn't have to take every day, but you can give maybe once every couple months or once every year or something like that. So again, the idea really is that over the insurance, almost 50 years, therapy for HIV has continued to improve. We did not stop when we had effective therapy. Science and research has continued to make even better therapy that has produced better results. I think all of you remember 2001. Everybody remembers 2001 as in the midst of the pandemic. It was a bad time for everybody, but it wasn't all that bad when you think about it from an HIV standpoint, because that was really the time in 2001 is when the FDA approved the first extended release injectable drug for adults living with HIV, in other words, for treatment. But by December of that year, the FDA also approved the first long-acting injectable for HIV prevention. And long-actings are not new to medicine. We have seen them in multiple other situations, for example, for birth control or for psychiatric medications. And long-acting agents are useful because they improve adherence, they're convenient, and they really give individuals a choice. Do you want a pill? Do you want an injection? What do you want? So where are we in people acquiring HIV globally? Well, the thought initially was when we treat everybody, there's not going to be transmission. But unfortunately, we're not yet able to treat everybody, and many people who are infected don't know it, and many people who know it are not necessarily taking appropriate therapy, are either not in treatment because they cannot have it or they're not able to continue it. And as a result of that, new infections have continued. They have clearly decreased since 1996 when we started a highly active antiretroviral therapy. You can see that the number of people dying of HIV and the number of people infected with HIV has gone down continuously from, the new people acquiring HIV has gone down from over 3 million, almost 3.5 million individuals, to now down to 1.3 million. But the reality is we really need to get below the number of people that are dying. In other words, we need to get down to 500,000 and we're new infections per year, and we're nowhere close to that. We're still far from that goal. So what ends up happening is you have 600 people dying, you have 1.3 million new infections. So the number of people living with HIV globally actually continues to go up. A similar thing happens in the U.S. In the U.S., we have seen down the numbers, overall, there's a decrease in new infections from, in a significant way. We're down to 31,000 in new infections per year in 2002. But the goal was to decrease new infections to under 10,000 by 2025 and under 3,000 by 2030. Goals that, again, we're not going to be able to achieve where we are. And new infections have gone down. They've gone down from 36,000 to about 30,000. And this declines really have occurred because of reductions in new infections in what's called the HIV epidemic jurisdictions. These were jurisdictions that under the Trump administration, under the first Trump administration, resources were dedicated to this jurisdictions in the U.S. that has significantly high number of new infections to really try to decrease the new infections. Again, showing that by putting resources and putting in creativity, we were able to do this. But again, we're far from the goal of getting under 10,000 new infections. What has also happened as a result of this is that the drugs we use for treatment are the drugs we use for prevention. Prevention is treatment. So we've gone into what called a serostatus neutral approach to HIV prevention, in which you go to the center of this, you get HIV tested. If you test positive, you go the treatment pathway. If you test negative, you go the prevention pathway. But at the end of the day, the idea is that you treat and prevent the disease on everybody. And if you're able to do this, you will be able to control the disease. And this will be really unique because you will be able to control HIV infection in the absence of a vaccine, something that many of us still believe that is not possible. So what has happened in HIV prevention? Well, clearly HIV prevention has gone a long way. We've gone a long way from, the dates of condoms and abstinence and be faithful, the ABCs of prevention, to now having drugs. And you can see we now have drugs that could be a single pill for HIV prevention. Tenofovir and tricidabine. This was approved in 2012. We also have injectables, and globally we also have even a deprivating ring for women. So what is the injectable? The first injectable used for prevention is the injectable called as cabotegravir. This is an integrase transfer inhibitor. It's very analogous to dolutegravir, which is the drug we use mostly for treatment globally. It's nanoformulated. It has a low solubility crystalline drug in an aqueous suspension. It was developed by ViiV Healthcare and is used in combination with injectable rilpivirine, another drug for treatment. But for HIV prevention, there have been 2 HIV prevention clinical trials under the HIV Prevention Trials Network of the NIH that compared cabotegravir injections to 2 months of daily tenofovir and tricidabine. And this study showed it to be very effective in men who have sex with men, transgender women, and cisgender women. So it was a safe drug, it was effective, and it really dramatically improved adherence because people came, got their injections, and left. You didn't have to worry about people taking a pill. And you can see here the summary of these 2 clinical trials. And again, I was the investigator one of the lead investigators in the HPTN 083 and one of the many investigators in 084. And clearly, these were, sort of game-changing studies. The initial trial, the HPTN 083, was designed as a non-inferiority design. And it was really interesting because at the end, it ended up being that it was a superiority result. We ended up having cabotegravir being superior to the control. And it significantly decreased the number of new infections. Same thing happened with HPTN 083, which was a study done in Africa, primarily amongst cisgender women. And again, among women, really nothing had shown. Oral therapy had not shown to be effective. And you can see here that cabotegravir reduced significantly by 89% the risk of new infections compared to oral tenofovir and triazolidine. So again, two studies that shown a superiority of cabotegravir, efficacy, and really were game-changing studies. Now we have, they're not perfect. You can have breakthrough infections, and these breakthrough infections occur at different times in both arms. Could be because of non-adherence, but could be from other reasons. And one of the concerns is that with cabotegravir injections, detection of new infections is challenging. You frequently may have an antibody test, but an RNA test that is somewhat negative or even a very low level. And in some of those individuals, you are able to detect integrase resistance, but not in everybody. And you can see here in this example that when the concentrations of cabotegravir, when the person got infected, when they first have their HIV positive, the concentrations of cabotegravir were right at borderline, were right at a reasonable amount. But then as the person got infected and became positive, you can see when they had their first positive test. And unfortunately, this person continued with cabotegravir injections. But again, you can see that their antibody tests were very difficult to interpret, right? They had a positive antibody test, but a negative or an indeterminate viral load. And even when they had a positive viral load, it was less than 40 copies. Now, from a global perspective, cabotegravir is very hard to produce. It really has not had the global impact we would have expected. Even in the US, hasn't had significant impact. So you can see that really it's been a significantly slow uptake. This is data from the US, and you can see that, despite having cabotegravir approved by the FDA approval is not the only thing you need. Only 2.5% of prescriptions for PrEP in 2024 were actually cabotegravir. Here in the... Oh. Can somebody turn off the microphone? Can other people turn off their microphones so we don't have echo? And you can see cabotegravir really has not taken off as much as we would have liked to in our country as well. Now, globally, we have to get this drug to people, but, there's issues of cost, of delivery, of testing. So a lot of issues have made this not be the drug that we expected globally. Now, prevention, PrEP is important. And this is a study from my colleague, Patrick Sullivan, presented last year in 2024 at CROI. And you can see here that as the number of PrEP users has gone up, we have seen a decline in new infections at the population level. But this is particularly significant when you look by states. In the states that have had significant expansion of PrEP, there's been a much greater reduction in new infections than in states that have not had a significant expansion of PrEP. So in other words, more PrEP in a population decreases significantly the number of new infections. And again, not all states have done the same job, the same work, and that then leads to different results. You get different results depending on what you do. So long-acting agents, long-acting cabotegravir is effective, but implementation issues continue to be a problem, and the detection of new infections is difficult, and this drug really has not had the global impact we all would have expected at this point in time. Now, the CDC has, and the U.S. Preventive Task Force has expanded PrEP, has said PrEP really should be available to all sexually active adults, and that's a grade 3B recommendation. And in people with high risk of HIV infection, they should be on PrEP, and that's a grade A, so a very strong recommendation from the U.S. Preventive Services Task Force. Now, a game-changing thing happened with the development of another drug, which is lenacapavir. Lenacapavir is a first-in-class capsid inhibitor, and it was approved for use in high treatment experienced persons with HIV who have multi-class resistance, and it showed to be effective in those settings. But the question was really, can this drug be used for prevention? And the bottom line is, yes, it can be used for prevention. And in fact, the efficacy of lenacapavir prevention was so much higher than in Purpose 1, the first study that I'm going to talk about. There were 0 new HIV infections in cisgender women in Africa, 0. And in Purpose 2, which was among men who have sex with men, there was only 2 infections in the lenacapavir arm. So again, this drug was called the breakthrough of the year in 2024 because of what it meant, right? What it really did to show a drug that literally could prevent, if we can get it to people, it literally can prevent them from getting HIV. Purpose 1 was done primarily in South Africa, but also in Uganda. And Purpose 2 was done in the U.S., Mexico, Peru, etc. You can see other countries. But this was a very diverse population globally of both men and women. And as I said, in Purpose 1, there were 0 new infections in cisgender women receiving lenacapavir. You can see compared to the people getting tenofovir FTC, there were 39 infections, and 16 infections in tenofovir FTC and 39 infections in those taking tenofovir TAF. And you can see in the Purpose 2 among men who have sex with men, 2 infections in the lenacapavir arm versus 9 infections in the tenofovir FTC arm. So again, a significant reduction in new infections by using lenacapavir. So this is incredibly exciting, and this again shows significantly superiority, and lenacapavir being 100% effective in PrEP among women, 96% effective among men. And this has given a new hope in HIV prevention that this drug, which can be administered not every 2 months, but every 6 months, or maybe even every year, can really be a game changer in HIV prevention. And a lot of us are very excited about this drug really taking off globally and being a game changer. This may be the drug that actually gets us to the decrease in new infections that we're all hoping to get to. But again, the issues, like anything, is going to be around implementation. Can we get the drug to people? Can we make it available? Can we make it accessible? Can we make it affordable? So here's a summary of the 2 studies. I'm not going to go into details because I already mentioned them. And again, both studies are published in New England Journal of Medicine. I would encourage you to read both studies. I would consider them some of the most important studies in HIV prevention published in the last year and a half or so. Lenacapavir, and those 2 studies was given every 6 months, but there's data from pharmacokinetics that maybe you can give it every year. And if that's the case, think about having an HIV prevention injection that you can give once a year. That would really be, again, a significant advance and will make HIV prevention so much easier. So there's a big pipeline of drugs being developed on HIV prevention. Lenacapavir is amazing, but it's not the end all. There are other drugs. There's a monthly oral PrEP being developed by Merck. There's combination studies. There's injectables. So really, the pipeline of HIV prevention is fairly significant. And again, a lot of where we're getting to is because of research being done within the HIV clinical trials networks sponsored by the NIH. So we have many options now in biomedical prevention. We have gone from, for some people, pills, daily pills are an excellent choice. For other people who struggle with adherence, long-acting antiretrovirals is superior to oral antiretrovirals. And patients who are on PrEP rarely acquired HIV. This is particularly true with lenacapavir. And PrEP works, and injectable PrEP works even better. So we have the tools to significantly reduce the number of new infections locally and globally. But the question obviously is, do we have the will? Are we going to put the resources? Are we now going to take these findings from research and scale them up to really benefit populations, both in the US and globally? And this is really, again, this is an old graph, but one that I like to use frequently. Confronting AIDS is about, prevention, treatment, and care. And it's all grounded on basic and clinical research. And I pulled this Science magazine from back in 2006 because 20 years later, in 2026, we can say the same thing. In this article, 25 years of HIV/AIDS, Dr. Fauci said, history will judge us as a global society, but not how well we have addressed 25 years of HIV as much as what we have done in the first 25 years. And I say the same thing today. it really is, we have the tools today in 2026. What's going to happen 25 years from today? Are we going to be able to end all new infections, or are we still going to be struggling the way we are today? Because really, it is a matter of implementing what we have in our hands and taking HIV prevention to the populations, to the people that need it most, and end new infections with HIV. And I want to end by acknowledging my two colleagues, Colleen Kelly, who was the PI for the Purpose 2 study, and Mary Laura, who runs our PrEP clinic at Emory, for facilitating some of the slides I'm presenting today. And with that, I'll end my presentation and turn it over to Dr. Coffin.
Dr. Arias, you're muted. And here comes Dr. Coffin now.
So we've heard, and now we turn to what is the virus doing? Dr. Coffin.
Thank you very much. I'm just trying to get my slides up here. I'm not sure what I'll maybe go back to here. Let's try this. That's not me. Where am I? There, of course. Ooh, like that. Start over again. There it is. Okay. Thank you very much, Wynn. It's always, as you point out, a great pleasure to join you in these. This is the first time we've done it on Zoom, for me at least, and so, but it's nonetheless a pleasure. Although we won't get to have our the meal together that we usually have afterwards. So I want to follow up on the great talk you just heard from Carlos Del Rio, to go back and drill down onto sort of one of the fundamental problems in dealing with HIV, and that is the fact that that HIV is, as was pointed out, HIV infection is by current standards is untreatable, in the sense that although a medical miracle really of the last century was the discovery of antiretroviral drugs that in combination could completely suppress the virus replication in infected individuals, it is not curative, and as soon as the slide shows, as soon as you remove the as soon as you remove the treatment, take an individual off of PWH, as we say, a person with HIV off of AIDS, off of the antiviral drugs, the virus, within a few weeks, the virus returns to what was pretty much its former levels. And it's done so despite being undetectable on therapy for all of these years, and for decades, perhaps. And the virus will nevertheless return to pretty much the same level it had before. Now, the level of virus activity, the replication in individuals, measured by the amount of virus RNA in blood, which is a very good instantaneous measure of the number of cells, productively infected cells, that are present in the individual at that time. Sorry about that. I think the mail has come. And by standard clinical assays, the virus seems to have disappeared completely into what you may well know is an undetectable level. However, if you use a more sensitive lab assay, you can almost everybody, you can see significant levels, very low levels, but significantly detectable levels of viremia, virus in blood, which, and again, for decades even, and as soon as you stop therapy, almost certainly it's those little levels of virus that return that can go on and infect a new cell. New cells are able to resume their infection and go right back to where they were when you stopped. Although the level of virus is undetectable, the level of viral DNA, which is the replication intermediate in retroviruses, is viral DNA, and principally in CD4 positive T cells, helper T cells in the infected individual, that has dropped significantly, but it remains at quite a higher level. It's only gone down perhaps to 1% or so of where it started. Perhaps as much as 10% of where it started, and just stays that way all the time. And this is representative of what we refer to as the HIV reservoir. Some fraction of this viral DNA is completely intact and is able, although it's not expressed well here, is able to, from time to time at least, generate new virus, which gives you rise to this very low-level persistent viremia, which can then, in the absence of therapy, can rebound and restart the infection. So what is the nature? What I want to focus on now is what the nature of this virus DNA is, what it's doing, and what the consequences are to the individual infected person. So I just have to this is just to remind you, I'm certainly not going to go through the whole replication cycle, that HIV is a retrovirus. The critical step in retrovirus replication, and the unique step that distinguishes retroviruses from literally everything else, is the ability of the viral DNA, once it's made by reverse transcriptase, to integrate at some site in cell DNA. Now, there's very little sequence specificity to this integration. The human genome contains many millions, probably tens of millions at least, of possible integration sites. But what there is some restriction on the types of genes that it goes into, and I'll get back to this a little bit later on. That provirus, when present, then behaves just like a cellular gene. It serves as a substrate for transcription, which is then translated to give rise to the viral proteins, which can then be assembled to make viruses. That transcription process, before I get on to that, very rarely this replication process does not go on this way. Instead, it stops, leaving the provirus in what we call some kind of a latent state, in which it's still perfectly intact in there, but it's not able to be transcribed into new RNA, new virus. By being latent, it also implies that it has the potential to be reactivated at some point by various kinds of signaling, and which we don't understand very well. But I'm not going to go into the mechanism of latency. That's another topic for another talk. But what I will go into is what the effects of the nature of this proviral DNA that you see and its effects on the cell. But first I want to talk a little bit about expression. Expression of retrovirus DNA is driven by signals in what's called the long terminal repeat, a sequence of a kilobase or more identical at either end of the genome. From the way that it's made, it has to be identical. That contains complicated combinations of transcription factor binding sites that plug the viral DNA very strongly into the cellular transcription machinery, so that it looks to the cell like it's a very good gene, especially well designed for picking up signals that are specific for the activated T cells that are its principal target for replication. And so it's the signals in the LTR that are that really direct what's happening as far as retrovirus expression and replication is concerned. And I want to raise one other issue now, is that the proviruses, intact proviruses in infected individuals that are capable of making complete virus, contain all the viral genomes, are actually relatively rare. Although most of them are intact at the time the cell is infected, there's a very strong selection right away for cells that do not have complete genes, and they may have the proviruses that do not have complete genes. They may have lost one gene or another, or they can be deleted altogether by recombination between these two identical sequences, leaving one of those sequences behind, but getting rid of all the genes. Those are very common in individuals that you can see, particularly on long-term therapy. And you can also see cases where there's just small deletions infecting this region of the genome or this region of the genome, and these are necessary for signaling and splicing. So their deletion or loss affects the ability of all of the genes to be expressed. And so these are actually the presence or absence of these genes has been turned into by the Silicano lab for what's called the intact provirus detection assay that I'll mention a little bit, in which you just look for specifically these specific sequences using very sensitive droplet PCR methods, and the loss of one of these or another, no matter what's going on with the rest of the genome, implies that the genome is likely to be incapable of expressing anything. Even the GAG gene, which does not require splicing, but it does require a signal here to be for the RNA to be translated. So another point I want to make is that LTR-directed transcription can also affect surrounding or neighboring genes, and that can give rise to some important phenomenology, including cancer occasionally, that I will discuss. So I'll give one technical bit, and that is, how do we define the integration sites? They're quite rare, only about one, only at most a few percent, and more often in individuals on therapy, maybe 1 cell in 1,000 to 1 cell in 10,000 actually carries the provirus. So to detect that provirus and determine where it is in the DNA, which is what I want to talk about, you need a very sensitive assay. And this assay is based actually on something that was originally developed by Charles Bagum in England for another retrovirus. But what you do, just very quickly, is you ligate on a specific DNA sequence, which will serve as a basis for priming with one end of the DNA, and you ligate that onto DNA, cellular DNA from a patient, from a person with HIV, that is that before you do that, you shear this randomly. That puts breakpoints. That breaks the cell DNA at random positions, and you then add a linker to each end of this viral DNA, and then you amplify this with a PCR using a primer that comes from a sequence in the long terminal repeat, so you know that that's viral, and a sequence that comes from your linker, so you know that you're picking up, in this case, the identity of the cell DNA, so you can see the integration site in the Illumina sequencing, the high throughput, high capacity sequencing that you do on the eventual product. And you can see where the integration site of integration is, and then you can go back to your gene maps and identify the gene. And then you can see where the DNA was sheared way back here. And from that, because you can only shear a DNA molecule once, of course, and only one DNA molecule comes from one cell, you can actually estimate the relative amplification of cells that had exactly that same integration site over time, since they were infected. So cells that have grown up into clones will show multiple breakpoints for the identical integration site. And so some of the slides you'll see this assessed as the number of breakpoints, and that's what I'll mean by that. That's a relative measure of the number of cells that were generated by replication of that one cell it started with. Now, the virus HIV infects a cell, CD4 positive T cell, whose job is to respond to antigen and grow it up to large quantities. And so, as that cell grows, the provirus that's in it will also grow along with it, along with this integration site, and you'll generate more and more breakpoints. So when my colleagues at the time at NCI Frederick first ran this assay on an individual we referred to as patient 1, it's no longer politically correct to identify individuals as patients, but patient 1 we named him patient 1, and he preserves that name as far as I'm concerned. So the first such analysis gave us, it really was to us a big surprise. And that was that very much half of the DNA here in this pie chart was in large expanded clones. The largest of which, or shared largest of which, was in a gene that we identified. It was in a gene, but we couldn't identify exactly which gene it was because it was in a gene that was highly repeated. We believe a ribosomal protein gene. So we couldn't tell exactly where it was, but it knew where it was. It wasn't important, so it wasn't as important to us to know where it was. But this, and as it turned out, this gene was capable, was completely intact. It was making infectious virus. And this person first came to our attention actually because he had detectable amounts of virus, despite being on fully suppressive therapy. And that virus was not resistant to any drugs. It was made because the clone of the cells was rather productive and also had grown to a very large size in that individual. So given the sample size, we could, we could estimate that the clone, I'm sorry, given the sample size that we can assay, obviously you can't assay all of the cells in an individual. We can only assay at best about 1 in 100,000. So to see a clone means to see a clone of 2 cells means we must have had at least 200,000 cells in that sample. And in this individual, we had a lot more than that. So this is coming from a very big clone of cells that were all, no doubt, all responsive to the specific antigen. We did not know, we had no way of knowing at this time what antigen these cells perhaps, or what was driving the clone. I will give you arguments that it's really, all of this clonal expansion is driven primarily, if not only, by the natural response of these cells, host cells to antigen. I'll also draw your attention to another set of sites. These are all different sites, A, B, just indicate different sites, but all in the same gene, a gene called MKL2. It's now called MRTFB. And on this short list, no fewer than 4 of the most prominent amplified sites in this were in this particular gene. Again, indicating there's something special going on there. And I'll come to that in just a minute. In fact, I'll come to that right now. So a big clue to that comes from when you look at the distribution of integration sites within that gene. So this is the MKL2 gene here. We also compared this to just taking freshly isolated CD4 cells from an individual, uninfected people, just in normal cells, and infecting them with HIV, activating them, infecting them with HIV, and then after a couple of days determining the integration site distribution, is what you can see is that the original genes, this almost certainly reflects what was happening in patients as well at the same time, and in people with HIV at the same time, taken at around the time of infection. However, after a long time, most of these cells died off or just were not within our sample. However, very prominently, there's a series of 3 small introns here. And altogether, these only amount to 7 kilobases, which is about the size of a provirus. And all of the integrations in this collection of individuals we're looking at here were within these 3 kilobases. And remarkably, you'll notice these proviruses, you can tell by the arrows, these proviruses are oriented in either direction relative to the gene. All of the proviruses in this individual on therapy were in these 3 introns, and were pointed in the same direction. I'll leave it to you to calculate what the probability of that is just by chance, but it's very, very small, and clearly there's something going on here. MKL2 is not the only such gene. A prominent and the best studied one is a gene called BOC2 that I'll talk about a little bit more, but exactly the same thing happens, and that is to say, you see in this case, they are within an intron of the gene, and they are in a position where they could direct, position just upstream of the translation start site of that gene. The RNA starts here and goes this way in this particular gene. And these are all pointed in the same direction, again, in all the same direction of the gene, and are likely to be affecting what's going on here. And this is not a new phenomenon in retrovirology by any means. There are many genes with lots of viruses. These are all viral vectorology examples, but it's been known for many, many years, since about 1980, that retroviruses can cause cancer by this insertional modification of gene expression, which I'll refer to it as, where a provirus, particularly one integrated upstream of a gene or near the beginning of the gene somewhere, the long terminal repeat of that can take over the expression of that gene and can cause a gene to be overexpressed or to be expressed in the wrong cell type. And in many cases, in a number of cases, actually hundreds of cases are known now, of genes are known now, where that can lead to cancer. And in fact, the study of these genes and this mechanism of cancer were, in my opinion, the most fundamental breakthrough in our understanding of the basis of cancer, going back now over 40 years. So, and I'll point out that the genes do not need to be necessarily even in the same orientation of the provirus, because although the LTR can directly promote expression of a gene, proviruses in the opposite orientation can also direct expression of the gene by enhancer activity, where enhancer sequences within the LTR can activate a promoter nearby. And as in this example here, and as in the case of integration of a gamma retroviral murine leukemia virus-based vectors into the LMO2 gene found in patients with treated with a very early form of gene therapy for SCID disease. So, in the particular case of interest, there are three prominent genes. There are more. I'll give you a list in a minute. Where we can make a guess as to what's going on. I say BOC2 is very important in the immune system, and I'll mention that later on. MKL2 is a developmentally very important gene in maintaining structure of organs and particularly the vascular system. How it's involved here, we don't have a good idea. STAT5B is important in immune regulation. And these are the three most prominent genes that have this property where you see these very clustered integration sites. And in the case of MKL2, it's particularly interesting because in MKL2, what actually that leads to is a truncated protein that's removed, particularly a negative regulatory portion of the protein. So that likely not only increases the expression, deregulates the expression, but also probably reactivates the gene product by relieving it from regulation. We don't know much more about what this gene does. Unfortunately, it's a very interesting one. So, and all of these genes, these are frequently referred to. If you read the literature on this, you'll see these referred to as cancer genes, because all of these genes are directly or indirectly related with cancer in experimental models. However, and that led to the hypothesis that they might have been driving the clonal expansion that you see, and possibly also cancer that you see in HIV-infected individuals. However, there's no specific association of proviruses integrated these with either cancer or preferential growth of the cells that carry them. In fact, it's much more likely, and as has been very nicely shown by a paper again by Francesco Simonetti in the Silicano lab, that the clonal expansion the dramatic clonal expansion that you see is in fact not unrelated to where the provirus is in fact driven up probably entirely by the normal antigen-driven expansion of a clone that is a T cell clone that is reactive to that antigen, that is killing cells that express that antigen and grows like crazy to accomplish that. And then when its job is done, dies off rather rapidly, almost entirely. And but you'll notice, and these are the large, what Francesco was able to do here was to identify the antigen that was responsible for stimulating these rather large clones from these patients, and to identify then the specific T cell receptor in these clones that marked the clones as in the same way the provirus did. And so it was able to make the correlation between the T cell, specific T cell receptor and the expansion and the provirus. And they but a remarkable feature here of the clones that he saw was the frequency with which the integration sites were in one of the genes that I've just been discussing, BOC2, MKL1 in that case, a paralog of MKL2, and STAT5B. There were 7 out of 32, or nearly a quarter of the large clones had these genes being in a position where they could be activated for expression by these proviruses. If you look at the population of overall, which we did some years ago, you'll find that only about 2% of the total gene integration sites are in this collection of genes, and the few more that we've identified by this process. That suggests rather strongly, I would say, although the numbers are not large, that there is some kind of synergy between these two, between the expression of these genes and antigen stimulation in the process of clonal growth. And Francesco pointed out that in different cases, the amounts of cells that are infected within a given clone. So you could identify the size of the clone by the T cell receptor sequence, and you could identify the ones that are provirus by the presence of the provirus in those clones. And sometimes, essentially, all of them were infected, where she interpreted as being an infection, rather very near the beginning of the time of growth of the cell, which would have been the first appearance of antigen. The dotted lines here are antigen expression. The orange is infected, and blue is uninfected clone of the same T cell receptor. And sometimes they were very small, as in the case of this particular clone. Okay, so So he portrayed it this way, which is a very nice way to do it, except that I take somewhat issue with what's going on here. The synergy implies, what I've told you before implies that the proviral insertions are not, again, driving the expansion of these clones, but I would hypothesize that what they're doing actually is preventing the death on the downside. When the antigen goes away, these cells die off rather rapidly, but they leave behind some memory cells that are around to kick off the immune response when the antigen reappears. So from those memory cells, now you have a reservoir of cells that's ready to go. We all carry very large numbers of memory cells to all kinds of antigens and to things we were vaccinated for many, many years ago, and all kinds of infections we've had, perhaps things we've eaten, who knows? But they're ready to jump out and protect us. The reserve is ready to jump out and protect us when the antigen reappears. However, what I would argue is that it's much more likely that the integration of these specific set of genes has that, for one reason or another, perhaps has the property of reducing the level that the extent to which cells that carry that die during this phase. And with BAC2, actually, there's good evidence for that. So what I would argue is that this pattern actually looks more like this, where the infection is back early here. T cells prefer to replicate, proviruses, HIV refers to replicate in activated CD4 positive T cells, which is what would be represented by the effector cells in this category here. If it's an infected cell here, if it doesn't give any kind of it doesn't contribute any preference to it, it's one cell in a million maybe, and the probability of it surviving to grow out, it would be very small. But if in fact it contributed something like reduced death rate to the virus, then you might imagine that you get this kind of a pattern where they grow about the same at the same levels they would, but the more and more they die, they die less. So there are more of them around every generation than the ones without the provirus, and by repeated cycles of this process, they take over the clone to a greater or lesser extent. So, so that's my argument. So what might be the mechanism of this? Well, for most of these genes, we don't really know. But let's go back to BAC2. So when I had a discussion on this recently with Yachi Ho, who's an expert on BAC2, and after our discussion, going back to BAC2, she asked her AI program to generate a slide about on the theme of back to BAC2, and so that's what the AI came up with. You got a different BAC, and but going back and forward to a more modern way of playing the music that he wrote, so many years ago. But you also have a theme here from Back to the Future, and so that's AI's and Yachi's contribution to this talk. But she also has done very nice studies on her lab is also at Yale, has also done very nice studies on what's going on with BAC2 generally. This now is the expression of BAC2 not driven by HIV. These are in cells. These are in HIV-infected cells, but that were isolated from the gut. And what she found was, without going into detail, what she found was that BAC2 expression really is a master regulator of the survival and growth of cells that come from effector memory cells, most of which die when the antigen goes away, but a few of which go into this resting memory state. And BAC2 expression is very important for that. So BAC2, if the provirus promotes expression of BAC2, you could argue that it might be what it might well be doing is causing cells that contain it to have much greater probability of going into this state, which is now ready to be reinduced when back into effector cells if antigen reappears. And so that with repeated cycles of this process, these would build up and up in the way that I showed you. We don't know what the products of the other genes that have been identified in this way. There are another 6 genes. I mentioned MKL2 and STAT5B. STAT5B also has many functions in T cell response and survival. So you could, I don't know exactly how this would work for that gene, but it's not unreasonable to expect that it would, that its overexpression and continuous expression would have a similar effect. Others we really don't know. It would be interesting to get some more experimentation going to find out. So, some years ago, a student in my lab, John Yoon, back in around 2000, was interested in seeing whether he could replicate this process of clonal selection simply in vitro culture, reasoning that a provirus is that infected a cell in such a way that that cell was now grow a little bit better than the uninfected cells. That is to say, it was in an integration site that made it possible to overgrow the other cells with time. What he did was to take HIV that was replication defective because the envelope gene was no good because it had been replaced with the GFP gene, which allowed us to track and select for infected cells. He infected a culture of freshly isolated CD4 positive T cells, passed those cells in culture under appropriate stimulation with IL-2, occasionally re-stimulating with beads that mimic antigen expression that would allow the cells to stay alive. Otherwise, they will tend to die off the same way antigen-missing cells would in vivo. And so it kept them going now for about 2 months. At intervals, then would extract the cells and run run an ISA, an integration site assay that I mentioned before, looking for the location and the relative clonal expansion of any given cell type. And he was first thinking that if one of these genes if these genes were responsible for directing overgrowth of these cells, you would expect the numbers of breakpoints in those to selectively increase with repeated analyses. If, however, something else was doing that, or nothing was doing that, you wouldn't see this effect. But you might find another gene. And when he ran it, sure enough, none of these cells showed any sign of overgrowth directed by integration in the gene. And however, another gene, STAT3, a very interesting gene, she did show such signs. And I'll just show you quickly what the data, some of the data looked like. Right after infection at day 3, you can see this shows the whole genome spread out over a single plot here. These are all the different chromosomes, 1, 2, 3, 4, et cetera. And this is now the number of breakpoints for integration sites in genes located at all these positions. And so you got thousands and thousands of integration sites. There was a pretty good coverage of integration over the whole genome. This is about 3 days after infection. So you saw some that are present twice in the sample, some that were present 3, and a couple were even present 4 times. And this just reflects cell division from the time that he started. So that a provirus in a gene that was where there's only one of in the whole culture would become 2 when the cell divided, and so on. And depending on how you sample them, you'll see some more or fewer of those. And decreasing numbers, of course, of the number of cells you're looking at. After 57 days, you see a very different pattern. You see there's still a lot of background of cells with proviruses all over the genome. However, you see this very impressive cluster here at chromosome 17. You also see some other singlets of about the same level of number of breakpoints. We're up to 60 breakpoints here. It's not insignificant. And where are all these proviruses located? Well, if you expand this out, they're all in the same gene here. That's the STAT3 gene. This is the STAT3 gene spread out. The All of the white here is introns. Exons are all these small regions between the colored lines. So actually, this is typical of most of our genes. Very little of our gene is actually exons coding information. We're almost all intron. And the integration of proviruses was very focused, again, all the same orientation, mostly in this particular experiment, in this intron. Repeat experiments have also given us collections of integrations with more proviruses than the one here seen here in this intron. You can see there's a large background of things that are present one-off, and these are obviously non-expanded proviruses. So, and these again are all in the direction of in the orientation of the gene. Okay. So these are obviously positioned to greatly enhance the expression of this, take over control again, the expression of STAT3. What is STAT3? And in this particular case, STAT3 is driving the expression of the cells in this in vitro model, whereas the other gene, none of the other genes we spotted for this do that. Will have that property. STAT3 is actually a well-known oncogene, particularly in lymphoma, whose mutation can decouple its properties. It's a gene that responds in signaling by IL-6, by the interleukins IL-6 and IL-10 through JAK kinase, which phosphorylates the STAT3. Phosphorylation does two things. For one thing, it causes dimerization of the protein, and for the second, it promotes the dimerization promotes transfer of the protein into the nucleus, where it's a transcription factor and now activates the expression of a large number of genes that are responsible for cell survival, proliferation, and mutations in this gene that decouple these events, or in the kinases that lead to it, are well-known drivers of cancer, especially lymphoma. So the question is, we know that among the among the AIDS-related malignancies, there are a number of lymphomas, particularly B-cell lymphoma, but also is most prominent, but also T-cell lymphoma, for which no opportunistic infection or other means is needed, has been identified. And but actually I got a bit ahead of myself. So let me first point out that that just to show you the LTR-driven expression, what it looks like. This is now through the same kind of sequence data was put through a program, an online program called Gene Viewer, which shows you, the same cells where DNA was, or RNA from the same cells was subject to RNA-seq analysis, randomly sequencing the whole genome in bits and pieces, the whole transcriptome in bits and pieces, and then comparing the infected cells from several clones of infected cells that we obtained from several different replicates with uninfected cells from the same replicates, and made possible just by selecting for GFP positivity. And you could see where the location, downstream of the location of the provirus, there was a dramatic increase in the expression of the mature spliced RNA of all of the introns, of all of the exons downstream of the provirus, as when normalized to expression of exon 1, which is upstream of the provirus. So exon 1 was the same in all of these. In the controls, the exon, everything was about the same. However, in these, the provirus is actually way off scale here. the provirus stimulated transcripts are way off scale here. So this is kind of a dramatic way to demonstrate the effect of the provirus insertion. So so is this involved in HIV infected associated lymphomas? So HIV, as I said, cancers are well associated with HIV, largely due to co-infection with more directly oncogenic viruses, particularly herpes viruses, Kaposi sarcoma, and Epstein-Barr virus, as well as human papillomavirus and probably a few others. That's where most of the malignancies come from. It's also been proposed and discussed, although the evidence for this, I think, is not so great. The chronic inflammation, which does also affect individuals on HIV, even on therapy, or possible chronic viral protein expression could also, at least in theory, be drivers of oncogenesis as well. I think there's less evidence, less direct evidence for this, less well studied, but still possibility. However, back in 2007, there was a paper published rather obscurely that we didn't see until we'd done the experiments I just showed you, and then it was easy to find, which reported in a case of B-cell lymphoma, which is the most prominent HIV-associated malignancy that does not have, some of it is due to Epstein-Barr virus, but a lot of EBV negative lymphomas do not have an obvious cause. And so they found that in at least one of these, there was an HIV provirus. It was really in exactly the same place in STAT3 as the ones in exon 1 of STAT3, as I just described. And they could show that was driving expression of HIV itself. They didn't go into details of the mechanism. So so that provided impetus for us to go and look. And we looked actually quite a few B-cell lymphomas. Unfortunately, we only had formalin-fixed paraffin-embedded DNA DNA from formalin-fixed paraffin-embedded samples, which is badly broken up and was not suitable for any kind of quantitative analysis or the required of the integration site, which requires good quality DNA. However, John Mellors at University of Pittsburgh, along with Steve Hughes at NCI, were able to obtain some T-cell lymphoma DNA. T-cell lymphomas are also increased in HIV infected individuals, but are much rarer. But they were able to get some frozen tissue samples, and were able to then to go through them, and they were able to find proviruses integrated again in intron 1 of STAT3 in exactly the same place as some of ours and the previous, and when we just discussed exactly the same general region. And I call your attention particularly to this provirus 12A because it tells us something interesting about the provirus, and that is there were two sites. This is the HIV-induced these lymphomas are cutaneous, so they had been able to get skin biopsies fairly easily, and so they took a couple from two different sites in this provirus, and they discovered that not only were there apparent activation by proviruses in STAT3, there were also provirus activation in a kinase, a tyrosine kinase. Remember, phosphorylation of STAT3 is necessary for its activation. And they found what's likely activation of expression of this kinase, LCK. But from two different sites, there were two different locations. And that means that these were two independent events. This is one event here. They were all the same. They all had exactly the same integration site here. But in this case, there were two different. So that implies that integration in STAT3 must have been followed by and perhaps simultaneously with spread of the originally transformed cells. So this STAT3 alone integration almost certainly must have done something to promote the expansion of these cells, which could then spread to other sites and then get infected again independently to bring up the LCK, the kinase. It was likely, this is not the one that usually activates it, STAT3, but it's capable of doing so, and likely allow the tumor to progress that much more rapidly at these two different sites. So one more interesting aspect that I'll go through very quickly is that another one of the proviruses was very defective. It had a big deletion that took out most of the 5' LTR, which is what usually directs provirus expression, as well as almost all of the POL gene, the reverse transcriptase and integrase, and and that they could show that expression was rather driven by the 3' LTR, right into just upstream of where the first coding exon, which is exon 2 of this provirus, of this gene, and so that could direct expression. But LTR directed expression requires the TAT protein, and that this TAT protein was incapable of being expressed from the LTR. But they found that it was indeed expressed not from the LTR, but from the normal STAT3 promoter, could drive the expression of this TAT protein, which could then support transcription of through the whole gene. So that that emphasizes the necessity for expression of the TAT for this kind of expression. And that raises the possibility that inhibitors of TAT, of which some have been developed in the lab, none clinically as far as I know, but if we had such inhibitors, they may serve as a valuable adjunct to treatment of this otherwise rather incurable and highly lethal disease. So, so much for all of the proviruses we've talked about so far, although they've had very interesting and important effects on the infected cells, and perhaps by the expression of partial gene products and so on persistent immune activation and inflammation that's typical even of HIV individuals on therapy. But so far, these proviruses are all highly defective. So, although they are potentially important for cancer and likely persistent immune activation, they cannot be responsible for viral rebound. And given the cytopathic effects of HIV and the very effective, actually, immune response against cells that are expressing HIV antigens, there must be some cells from that contain intact proviruses that must be protected from these events. And protection may be affected by simply latency of the provirus, where for reasons we don't completely understand is incapable of being expressed until it's activated somehow. Recent studies, so there's been more recently an effort to identify the integration sites specifically of intact proviruses. This is a challenge actually, because in such proviruses, if you do the arithmetic, only about 1 in a million CD4 positive T cells in your body, and if you're an infected individual on therapy, only about 1 in a million contains an intact provirus. So your work is cut out for you trying to both identify, so we can identify the proviruses, the structure of the proviruses. We can identify their integration sites, but it has been a challenge to figure out how to identify both at the same time. So several laboratories, including my former colleagues at the DRP, Sean Petro and Mary Kearney's lab, and the Lichterfeld lab has been most prominent with this, using a modified ISA protocol that I won't get into, has found in cells there are two principal types of intact of sites for intact proviruses. One is in satellite DNAs or deeply non-genic DNA. These are lamina-activated domains, which are specific for the nuclear surface of the nucleus, the inside surface of the nuclear envelope. And these are imagined, and probably are, in some state of deep latency, which express heterochromatinized that suppresses the expression of everything in its purview. and is essentially effectively and presumably it's effectively non-inducible. We don't know if these, I don't know at least if any of these proviruses have in fact been shown to be inducible. The other class of genes, quite remarkably, is a class of genes referred to as crab zinc fingers genes, also labeled, usually numbered as ZNF something. ZNF genes are a very large gene, about more than 400. In fact, the largest gene family in our genome. Contains a sequence-specific binding domain, the zinc finger domain, which is a bunch of little sequences identified that can bind very specifically to little short 3-nucleotide DNA sequences that are strung together in these proteins. So there are like 8 of them that can now specifically interact with very specifically with much longer sequences. Sequences are long enough to be unique in the genome. And so of these 400, these all vary in these zinc finger sequences so that they all bind to different sites. But in addition, they're fused to a protein, CAP1, which recruits a whole bunch of other proteins which are responsible for silencing the genome that they're in, either, for example, by kicking out acetyl groups from histones, which are acetyl groups here are activating usually, or adding methyl groups, trimethyl groups, for example, K9ME3 on histone 3, which are highly suppressive. They're also responsible for causing DNA methylation. And so these are proteins that have been rapidly evolving throughout our evolutionary history to both perform important regulatory roles in development and probably also even in differentiation as it's ongoing, and also prominently to suppress the expression of transposable elements to keep them from doing genomic harm. And they've been capable of rapid evolution in that regard by changing these zinc finger genes and just preserving the rest of this. Okay. Now, remarkably, this is an analysis that was done by Jason Rausch and Sharon Patro. If you look at it in vitro infection of these are targets. These are reasonably good. Some of them are reasonably good targets for integration in an in vitro system, where you see about 1% of the integrations are into zinc finger genes of this class, actually, that have been identified. And if you look at all sites for integration on finger, there's a slight enrichment of these, a doubling of the frequency of these in patients on therapy as compared to everything. But if you look at, these are the location of intact, okay. And if you look at zinc finger sites, however, and this is intact proviruses, you find that half of the proviruses, intact proviruses you see in this collection of a small set of individuals that was studied here, are in zinc finger genes. Remarkable for the statistical, the p-value for this is something on the order of 10 to the minus 16th. So it's really quite remarkable. So, so however, only, and again, only, but if you read the reviews and so on in this, there's often a confusion. It'll assume that all zinc finger genes have this property. That's not true. It's only a very small fraction, and of them actually do. And again, in this small study from the Miller's lab, you can see that one of these, even in 2 individuals, one of these genes, zinc finger ZNF 721, was hit twice in 2 individuals. And if you look at all of the zinc finger genes identified in this study, and then you ask what and you go to the literature, you can find 17 papers that also did the similar kinds of experiments. And what you find is that in these 17, all but 4 out of all but about 14% of the of the of the of the zinc finger genes identified to have intact proviruses were reported only once in one paper, and the remaining 86% were found by at least two groups in two different papers, and this data set was carefully curated to remove duplicate individuals who might have been seen in two or more studies. These are all independent individuals. These are all at different sites within the same. All the ones in the same gene were in different sites, and so these were clearly. This is clearly quite remarkable, actually. It's a very small subset. Remember, there are 400 of these genes. Thus, only a small fraction of these genes provide such safe sites for survival and expansion of proviruses capable of expressing virus on expansion. And indeed, in this particular individual, provirus genes with the same sequence, RNA with the same sequence, is actually found even in the untreated individuals, even in the treated individual persisting on therapy, persistent viremia, and that's because it's a very large clone, 2% of the total RNA, 2% of the total proviruses are in this particular clone, which is very large when you consider the number of proviruses. So it's been proposed that, as I mentioned, that this effect might be due to is based on the syllogism that the proviruses are found in very highly suppressive regions of the genome, like centromeres, and but they're also found in zinc finger genes. Therefore, the zinc finger genes must be highly suppressed to suppress the provirus. You can see the logical difficulty with that way of thinking. In fact, the assumptions of that seem not to be true. Crab zinc finger genes are often well expressed in the target cell population. Indeed, ZNF721, the one that I've mentioned the most, is the most expressed of these. In this same PWH set that was used for the previous slides, this is around 70 or 80 TPM, which is good, pretty good expression actually, way above average. And
excuse me, Dr. Coffin. Yeah, Dr. Coffin, we're just about out of time. I'm hoping you can wrap up so we can get to the Q&A.
I am just about to wrap up, as it happens.
Okay, thank you.
So the provirus, but also the original integration frequency of proviruses is strongly related to the level of gene expression, the rate of integration of this in the in vitro samples shows that at the time of integration, these cells were almost certainly expressing at a reasonable level that may be consistent with what you see here. Cells that don't express genes that are not expressed at all are very, very poor targets for integration. In at least one instance, the provirus in this gene is in a highly expanded clone and gave rise to detectable non-suppressible viremia. And we think it more likely that, as in all of the examples discussed previously, the effect of the provirus expression in the ZNF genes is not because the provirus is being greatly, deeply suppressed. It just seems not to be the case. It's instead because some effect, we don't know what, but some effect of the provirus on the expression of that gene is directly responsible for the ability of the cell to evade the immune response. And I will close with that statement and thank all of these individuals for their help in preparing this talk.
Go to the clinical ones. All right, thank you very much, both of you. As I pointed out in the very beginning, a bridge has multiple parts of great complexity, and we certainly were exposed today to both of these, both in terms of the translational aspect and the complications of global and other therapy, and certainly into the amazing complexities of the virus. Now, we have a whole bunch of questions, and so we're going to begin. We have a question for Carlos. One of your slides, it looks like it took about 10 years from 1996 to 2005 for the number of people living with HIV to begin to decline. Do you have any idea why that may have been?
Well, it took about 10 years for the number of people living with HIV to decrease the number of deaths. And that's because the time it took us to scale up treatment, right? You needed to be on treatment in order to stop dying. So that delay was a delay of, again, in 1996, we knew antiretroviral therapy delayed deaths, significantly improved the lifespan, but we did not expand it right away. So it took some time. Really, it was really PEPFAR and the Global Fund and all the world effort to get antiretroviral therapy to people that really dramatically changed the global pandemic.
Carlos, we have a very important question that's been asked by many people here. Here it is. At this stage, what are your recommendations on which organizations are most well positioned to lead responses to the current stage of the HIV epidemic? And what are some of the barriers based on your assessment?
Well, I mean, I think we've gotten this far by a combination of research, implementation, and research, I mean, pharmaceutical industry, NIH-funded research, researchers coming together, very important people with HIV. I think from the very early in the epidemic, people living with HIV have been part of the solution, have been part of the research agenda, have been really at the forefront of research. But then you need governments, right? You need governments locally and globally to really put on the resources. And at the end of the day, you need a local health system. You need really the way to get people in therapy. You look in prevention. You look in our country, we have a sort of patchwork of treatment programs. We have some states that have expanded Medicaid, some states that have not. We have the Ryan White program. We have a variety of different programs. Nowadays, for example, because of pharmaceutical and funding restrictions, it's a lot of problems sometimes getting patients in the right anti-prevention and the right treatment because you need prior authorizations. So really what you need is a system that works seamlessly. And again, at the end of the day, you need healthcare that is affordable, available, and accessible to people.
Excuse me, Dr. Coffin, you can stop sharing now. That way the three of you will be on the screen.
Okay.
Carlos, what is the current status of the epidemiologic factors in HIV? Is it the same population, more or less, as it was?
It depends when you go, where you are globally and locally. For example, there has been much in the United States. It continues to be a disease disproportionately impacted men who have sex with men. But because there has been a significantly higher expansion of PrEP among white MSMs, now you see a lot more infections in African American and Hispanic MSMs that have not had so much PrEP expansion. In Africa, continues to be an infection that primarily impacts heterosexual populations, but globally, we're beginning to see a significant increase, primarily in Russia and in former Soviet Union countries, but in other places, of injections among injection drug users, which have been really a population that has not had access to care prevention as much as other populations have. So really, the epidemiology changes depending on what the local situations and what the local epidemiology are.
What is the role of education, particularly in public schools and high schools, regarding HIV in the United States today?
Well, we need to continue educating people about the role of HIV, but we also need to continue educating people about, in Congress, and we need to educate people, the funders. I remember being in Congress and somebody saying to me, well, HIV is no longer a problem, right? And I said, well, we have 36,000 new infections per year. If you had 36,000 new Ebola infections per year, you would think it was a problem, right? And this person I didn't know there were 36,000 new infections per year. So we have to educate people in schools, but we also have to educate people in Congress. We have to educate people at all levels, because the reality is a lot of times I feel that AIDS and HIV is forgotten but not gone.
Here's another one for you, Carlos. Public health messaging has shifted over time from abstinence and condom use to treatment as prevention and undetectable being untransmittable. How do you think communicators should navigate these parallel messages so that it's understood accurately without people interpreting it as risk-free in all contexts?
Well, again, at the end of the day is we have to educate people of where we've gotten because of research, because of science. And we're in a very good place right now. We have a lot of good advances, and we need to continue to educate people how that science translates into benefits to individuals, right? And that, to me, is the key, is really doing that continuous translation and application of science that makes people understand that, we didn't get there by accident. We got here because of research. We got here because of many things. So need to continue emphasizing that to people that the advances that have occurred have not been an accident, have been the result of really an incredible, dedication to science and advancing really care.
So, John, we have a series of questions for you and some more for you, Carlos. But, John, here's one from your friend and colleague, Henry Levin, who asks, would vaccines have greater long-term efficiency if BOC2 could be induced in memory cells?
That's an interesting thought, although playing with that might have side effects that you might not like very much. So, but it's certainly an interesting idea to think about.
So another one from Henry Levin. Do you think that transition from STAT3 to LCK integration is an endogenous reinfection, or could it be that STAT3 integration predisposes to opening the LCK site?
That's a possibility, but I suspect it's not necessary to imagine that. I haven't looked, actually. I have the data. I should go back and look and see what how good LCK is as a target in uninfected cells. It may well be, and of course, it might do that indirectly, because the STAT3 alone might be stimulating the cells more to might be inducing a sort of a partial activation of the cells, and it seems to, and some experiments have been done in my lab. And that may then promote reinfection generally of those cells.
Carlos, here are two questions for you. One, what's the current status of PEPFAR? And the other question is, do people who are taking antivirals have an increase in lymphoma formation?
Well, let's start with the second one. I think we're seeing people with HIV live longer, and as a result of living longer, they start developing, cancers. Some of them HIV-associated cancers, but some of them HIV non-associated cancers, like lung cancer and esophageal cancer. I think they have a degree of immune. While they're virally suppressed, they still have a degree of immune dysfunction that makes them more susceptible to cancers, and that's why they need to be screened regularly. But lymphomas is not one in particular that they other cancers are also there. As far as PEPFAR,
I was going to say, I could add, they might also have a lifestyle that predisposes them, like
absolutely. as far as PEPFAR is concerned, PEPFAR continues to operate, but faces significant uncertainty. And it has a 1-year reauthorization, which is going to be expiring soon. And PEPFAR is trying to not only cut budgets, but also strategically shift towards a more in-country transition. Sustainability has been hard, and implementation has been hard with the defunding of USAID. But still, the government's still putting money into PEPFAR, and still, drugs are being distributed. There's been some challenges, but I would say PEPFAR is a little more stable now than when the Trump administration first took office, and there were a lot of cuts happening. Right now, we're trying to see a new PEPFAR happening, and this is almost a reinvention of PEPFAR. I think it's not necessarily bad. I think PEPFAR needed a reshape. It's no longer an emergency program, but we also need a plan, right? We need to figure out how do we get people continue to be on therapy, because we have a commitment, and we need to decrease new HIV infections, and for that respect, lenacapavir is obviously a big and exciting new drug, and if we can make lenacapavir more available, I think that will be good. But I think we also need quite frankly, to see a therapy sustained for those people that are on therapy, that we cannot stop their treatment.
Can I follow up with a question of Carlos?
Sure.
I was rather disturbed by some discussion I heard, I don't know, maybe 6 months ago, about the possibility of introducing sort of blanket application of lenacapavir to high-risk populations. And I think that's, maybe you'll disagree, but I think that's a very, very bad thing to do.
No, I agree 100% with you. I think, some people are seeing lenacapavir like the solution to all the problems. It is not. And I worry that we are overplaying that card. And again, it's something that I think it's important, but it's not going to end the epidemic. And expanding it in a way that will potentially could be dangerous, right?
Very dangerous, because lenacapavir is wonderful in terms of its potency, and its solubility, and its suitability for long release, and so on and so forth. But where it's not wonderful is its resistance. It involves resistance quite easily if you. So if it's given to an individual who's already infected but you don't know it, you might be able to improve that by doing RNA tests on everybody that you're going to give it to. But that seems like it doesn't. That's not in anybody's plan to do so. And saturation, particularly of high-risk communities, would almost certainly, I would say inevitably, lead to widespread spread of highly resistant virus, and lenacapavir no longer will be any good for anybody.
I'm with you, John. I agree 100%.
So I think it's a very dangerous thing to approach.
And that's why, I think it's a new tool. It's a useful tool, but it's not the solution to our problems.
So we have a question, John. When you see proviruses in the cells in which the HIV virus part is hiding, are there different proviruses in different cells of the same individual?
Oh, lots, yeah.
Do you observe any cellular heterogeneity in the expression of provirus, or in general, is it always the same provirus in a given case?
Oh, no. In the cases where the cell has clonally expanded from a single infected cell, yes, the provirus is the same, although it could, at least in theory, pick up mutations, but the mutation rate as a piece of the DNA is very, very low. But during the course of replication, that create all of the proviruses you see in all the cells. I mean, if you're an infected person, a person with HIV, you have on the order of, let me do the quick math here, Something like 10 to the 9th, something like 1 billion cells infected with HIV. This is in an individual who's on therapy. Every one of those cells has a provirus. Every one of those cells, if you strip that down, so you look at only the cells that are clonally expanded, you can cut that number in half. Maybe the average clonal expansion is 2, although it's a wide variance. You're still talking close to 1 billion different proviruses at potentially different sites in the genome. Now, there probably aren't as many as 1 billion sites in the genome, so occasionally a couple would be at the same site independently, but there'll be different proviruses. They'll have a different sequence. So the HIV accumulates a lot of genetic variation as it replicates. So during the course of the replication, while the virus is still replicating, you generate enormous genetic variation. It's much more than has been seen almost with any other system except for hepatitis C virus, which is even better generating mutation. So it's a very complex system in that regard. Everything is different.
So do either of you or both want to respond to the question that's been asked? Is development of a vaccine a lost cause?
I don't think it's a lost cause. I think that a vaccine is, we're learning a lot from trying to develop a vaccine, but more importantly, the reality is that we have we're learning other things from developing a vaccine, right? We're learning a lot of things from we are learning, the vaccines that we use for COVID were developed, quite frankly, because of research had been done by the NIH Vaccine Center trying to develop an HIV vaccine. So I think that we have an opportunity to continue advancing science and understanding better. We're using vaccines now for treatment. So I don't think it's a lost cause, and I still think it's a very important area of research.
John?
I think it's an enormous challenge. I would not go quite so far as to say it's a lost cause. I think trying to develop a vaccine along the lines that we think of traditional vaccines, I think, is a lost cause. But new strategies could well be arising that would get around that. The problem with HIV is the evolutionary problem. You have to look at the niche that this virus has evolved to replicate in. And it's evolved to replicate and survive in individuals who have perfectly functional immune systems and to stay there for decades in such individuals. And the wild ancestors of HIV do this, but they manage to do this in their natural host without causing disease. If we could figure all of that out, that would be also a very good thing to do. But that means that the virus is capable of maintaining itself and transmitting from individual to individual in the face of individuals with completely competent, fully competent immune systems, and it has many adaptations to it. So I have lots of slides on this. It's a question for another talk. I have lots of slides on why the sort of classical vaccinology with an inactivated virus or attenuated live virus is almost certainly not going to succeed. But there are other strategies, as Carlos points out. We're developing other strategies. There's a very interesting strategy of using so-called defective interfering particles or therapeutic interfering particles that are the virus itself with carrying a certain number of mutations in it so that it kind of outcompetes the existing virus, but it cannot itself replicate and cause disease. And there might be some that may well, it's obviously nowhere near development into a therapeutic strategy yet, but that may well, or a preventive strategy, that may well go somewhere, particularly in terms of possibly in terms of treatment.
So, John, if you could design an AI system that would be directed to answer any of the fundamental questions you brought to mind today, what might it be?
What might the question be? Don't ask me what the system would be. I know nothing about AI systems, but what might the question be? Is that what you're asking?
Yes.
Say it again.
Sorry?
Repeat the question.
Oh, I'm sorry. Well, let's go back from a different point, question. Where do you think HIV virus came from? Have we ever seen this kind of a virus before in an epidemic sense?
Retroviruses are not generally considered to be really good transmitters from individual to individual, but that, of course, is not true. There are viruses that cause spreading. There are a number of viruses that cause spreading disease in domestic animals. In chickens, there's some very dangerous viruses, and in sheep there are actually a couple of viruses that are quite dangerous. And so that's a new thing, but a virus that has the mode of replication and the properties of HIV has not been seen. HTLV1 is another. HTLV is another virus that retrovirus that does spread, and actually has caused quite a bit of mortality in some countries, among Aboriginals in Australia, for example, and also in parts of China. So that's also a very dangerous virus in that regard. Its association with the host is quite different from what HIV is, but it is a retrovirus.
But as we know from work done by many people that that is likely, HIV emerged like this as zoonosis, like most viral infections nowadays.
Exactly, yeah.
And I think what again, this human-animal interaction has been very important, and all the way from AIDS to Zika, these are zoonotic transmitted infections, and we need to remind ourselves that zoonosis really is how we're acquiring most viral infections nowadays.
That's right. In the case of HIV, it's very easy to argue that that the rate-limiting step was not the transmission of the virus from an animal. It was actually the conditions that allowed the initial spread of that virus in the human population. That has probably been happening for a long time in populations that were in close contact with infected animals.
The ability that the virus had to infect, and must say that it's not a very easy virus to transmit, right? I mean, we're not talking measles. We're not talking about highly transmissible agents. This HIV is hard to acquire, yet it has infected millions of people globally.
Right. A great many of whom know ahead of time how not to get infected, but still manage to do so.
Exactly.
Well, we keep hearing about stories from the public health arena that particularly young people who are treated with the antiviral
Uh-oh, I think he froze.
Yeah, we'll see. This is Chris. Okay.
People expanding.
Dr. Harris, you unfortunately froze through most of that question. Maybe you can rephrase it and then wrap up.
All right, John, we have another question for you. Is vaccine development, would it be possible for HIV patients with persistent viral load and zinc finger proviral integration? Would vaccine development be possible for that group?
I'm not sure why that would be a special group to target for that. It would be interesting to try to find some way of targeting viruses that were specific, proviruses that were specifically in zinc fingers, but because that might go a long way toward reducing the reservoir in people on therapy. But I don't know. I don't see what would be particularly special about the virus in that regard, in the regard to the possibility of generating a vaccine. That would be useful.
Well, we are running out of time, but I would like to conclude by asking each of you if you could perhaps briefly inquire of your colleague what you would like to see develop that would help you in your own understanding of HIV. Carlos, do you want to respond to that?
Well, I think that again, working closely with basic scientists and trying to get their discoveries into the clinic continues to be a critical component. And I must continue to tell young people and others in the audience that there's a lot of research to be done. There's a lot of things to be discovered, including, developing a vaccine, developing a cure for HIV. So there's a lot of opportunities to continue doing some very good science in HIV. So HIV is not over, and we need the work of everybody interested in science to get us, because we've gone this far through science. We're going to get out of this problem also through science.
John?
I would second all of that, and I would say that the HIV research history for the last 40 years, more than 45 now, since the epidemic sort of first appeared on our shores, or first became visible. It appeared well before that. Has been really remarkable in the back and forth interaction between very fundamental science down to structural biology and all the way up to the sorts of things represented by Carlos, public health and drug delivery and I think it's been a remarkable example of that should be a model for other much more of a model for other diseases than it often has been. So how you translate in issues, very scientific issues like the evolution of the virus in infected people becomes very, very important when you think of the spread of drug resistance and how you administer it and why you administer certain antiretroviral drugs. And without understanding one, you really can't understand the other. And that's been really a hallmark of the research in the disease, and it's been really a remarkable ride to be a part of it. A small part of that for the last 40 years.
Agree.
We have one other question I forgot that came in that several times, Carlos. You want to talk a little bit about cost with any of particularly the long-acting antivirals? How do you visualize societies are going to deal with it?
Well, cost is a problem, but let's not forget that when we started antiretroviral therapy at a global scale, the cost of therapy was considered to be insurmountable. And nowadays, you can provide antiretroviral therapy with 3 drugs to people infected with HIV globally for less than $1 a day. So the fact is the costs have gone down significantly. And the costs that we are seeing are not the real cost. And I think a lot of those drugs can be produced at a much lower cost. And it has been shown to be the case. So I wouldn't take, while cost may be an issue, it's not an insurmountable issue, because the fact is those drugs can be produced at a much cheaper cost in that we currently think about. And the example of treatment is there.
Well, we have run out of time, but I'm on behalf of, I'm sure, a large number of people on both sides of the bridge who participated in today's program. They share, as I do, really the excitement of these 2 brave new worlds of challenge in this persisting, difficult problem. So thank you both for your time, your effort, your presentation, and we greatly appreciate your taking part and educating us all. So thanks for
the invitation.
Okay.
You're very welcome as usual. Next time we'll have dinner, though.
Yeah, exactly.